Well, thank you so much for this opportunity to present our work. I'm very sorry that it has to be remote. I also realize that as the last talk, I'm between you folks and your own phage cocktails, or your own cocktails, I should say, except maybe for some folks in the back who might have gotten started early. But thank you very much for your attention. I'd actually like to start this talk by starting off with a different virus, HIV. Um, and maybe I'm dating myself, but I thought this conversation about, or this presentation about cocktails would be interesting in the context of thinking about the history of cocktails for this other virus. And I can recall, I think I was in school when the first single drug therapies came out for HIV in 1987. And then cocktails became available in 1995. And then by the time I was an infectious disease physician, or in my training, we had personalized three-drug regimens that were really about combining different therapeutic approaches and providing sort of orthogonal selection pressures on HIV. And where the field is now is essentially those regimens have gotten bundled. We now can actually give a single pill to folks with HIV, and we don't test individuals. We typically will treat and then monitor them for failures. And I think this is a really interesting model or a bit of history to think about in the context of phage therapy, because I would offer that phage therapy has followed a very similar arc from the perspective of development of single phages and cocktails and personalized regimens. But what I think we lack is sort of the conceptual infrastructure, if you will, for understanding how phages and antibiotics come together. And without that, we're unable to move through, the entire progression that heart has gone to the point where we have off-the-shelf phage therapy regimens. Nonetheless, I think that's an important goal, even in this era of really strong data around personalized phage therapy. And that's for several reasons. The first is that ideally, you want this to be high throughput and low intensity. You want phage therapy to be standardized so you can do things like optimize pharmacokinetics and formulations. There's obviously regulatory and financial incentives to having standardized off-the-shelf cocktails. And then, in addition, for many of the infections that I treat as a wound care physician, the infections end up being polyclonal. And so you're never going to have a full grasp of all the different strains that are present. So having broad spectrum off the shelf phage therapy cocktails would be really important. So the question we've been asking in our lab is it possible to develop broad spectrum off the shelf phage therapy cocktails? And that's what I'm here to talk to you about today. This is what happens when you drag your 14-year-old to a phage therapy conference when you talk about cocktails. You get little drawings like this one. So there are, of course, many approaches and strategies for cocktail design, far more than I could fit onto an individual slide. I think some of the ones that are worth mentioning or that we've been thinking a lot about are approaches that combine different receptors. But I think it's fair to say that most phage therapy centers rely on tailored cocktails that have been arrived at empirically and then tinkered with over time to yield good results and then optimized for individual patients and tested in that way. Later on in this conference tomorrow, former member of our lab, Dan Mueller, is going to be telling you about some work we've been doing where we were looking at the relative importance of bacterial receptors versus resistance mutations in terms of defining lytic phage host range. And as she'll tell you about tomorrow, our work comes down strongly on the side of receptors being the far more important consideration in terms of phage host range. And sort of inspired by this and some of the other work that I mentioned a moment ago, we started with the hypothesis that combining phages that target multiple orthogonal receptors can create phage cocktails with broad host range. And everything I'm going to show you here is work that was done by Kevin Kim in our group. And this paper is available on bioRxiv. And so we looked at Pseudomonas, and Pseudomonas has a bunch of different cell surface structures that are used as receptors by bacteria, including type 4 pili, flagella, and LPS, but then also others. And to facilitate these studies, we needed a quantitative rapid method for identifying or expressing bacterial killing so that we could do comparative studies. And so to do that, to provide that, Kevin developed what we've been calling a suppression ratio. And I think there are other things similar to this in the literature, but essentially looking at the difference in suppression you get over 30 hours with and without a given bacteriophage. And this is what that data looks like. And then we also developed what we've been calling a resistance ratio. So if you then take the phages that emerge from that first round and treat them again with the same phage, what sort of resistance do you develop? And this is that data expressed here. So for this isolate that's largely resistant, the resistance ratio, of course, is quite high. You can use these tools, these ratios, to essentially look at cross-resistance testing. And what we find is that phages that use the same receptors, and we went through each of the phages and clinical isolates involved to identify the receptor usage, end up being cross-protective. So all of these phages that use the type 4 pilus, if you develop resistance to one, you reliably develop resistance to all of them. And this is using a twitching assay that sort of brings home that point. So DSM-34, T4P, and Luz24 all use the type 4 pilus. You can then take the resistance matrix data that I showed you earlier, and you can put that together into a grid like this one that we call a complementarity matrix. And essentially, all of the phages here in the first exposure and the second exposure that are cross-resistant have yellow boxes, and then the ones that are susceptible come in gray. And what these data tell us is that for Pseudomonas, and we've looked at probably about 30 or 40 phages at this point, all of the phages that we've identified form 4 clusters of cross-resistance that correspond to the receptors that the phages in question use. I'm only showing 8 of the phages here, but you can expand this matrix to include many other phages, and that's what this data looks like. We then asked how many combinations of these complementarity groups do you need to make phage therapy work well? And we found that if you don't have any phages, the bacteria, this is looking at OD600, grow as you would expect they would. If you only have one phage or 2 phages from the same complementarity groups, you reliably get regrowth before 15 hours. If you have 2 phages from different complementarity groups, this will actually buy you almost 30 hours of protection. But if you have 3 phages from different complementarity groups, in many cases, you don't get regrowth at all or until much, much later. And I mention, bring up the HIV cocktails in this context just because of the, I guess, the magic nature of the number 3, I suppose, in the sense that orthogonal selection pressures probably have the same effect on bacteria in this context that antiviral drugs do in the context of HIV. We then looked at clinical isolates. I think this is about 12, and compared 3 phages that we've been calling the kimchi cocktail because the author is Kevin Kim, and this is cocktail number 1, so kimchi. And we find that each of the cocktails individually protects against some of these isolates, orange being more suppression, black being less suppression. But the cocktail covered these the best. And if we actually take polyclonal isolates, so combine different strains of Pseudomonas, PA14, PA01, and then a clinical isolate, we find that only the cocktail and not a mocktail made up of phages from the same complementarity groups suppress that polyclonal group of isolates. And so what we concluded from this is that the phage cocktails are effective against polyclonal cultures of Pseudomonas. We then asked whether this works for biofilms, and I won't belabor the point, but here again, we find that if we have 3 phages from different complementarity groups, they also work better for bacterial strains grown in the context of biofilms. And then we looked at whether these cocktails work better if administered simultaneously or in series. And we find that if we did dual therapy and administered the cocktails simultaneously, that suppression was better and that the resistance that emerged ended up being less. And so for both of these reasons, we think that dual therapy is better for these phages. We then developed what we've been calling a synergy score using, again, approaches that are out there in the literature, but here to take the suppression and resistance index data and to look at how well they complement conventional antibiotics that target Pseudomonas. So blue here is highly synergistic, red is antagonistic. And we find that some antibiotics, like meropenem over here, have reliably synergistic activity against all of the phages that we used in this group. Whereas most of the other antibiotics that we looked at, including aminoglycosides, tetracyclines, and fluoroquinolones, it's more heterogeneous and patchy. We thought this, again, is a helpful matrix because you can add phages or clinical isolates to it. And in our paper, we look at multiple other isolates and find very similar things to this. We then asked whether this works against clinical isolates. And we took 154 Pseudomonas antimicrobial resistant strains from our hospital. And we compared kimchi, our cocktail of 3 phages, here at the top, against aztreonam, which many of these bacterial strains are resistant to, against the cocktail of kimchi plus aztreonam. And what we found is that for 75% suppression, which was our cutoff for efficacy, the cocktail alone worked against over 80% of these clinical isolates. Aztreonam, in contrast, only worked against just about 40%. And the cocktail combination of kimchi plus aztreonam worked against over 95% of these isolates. And that dropped a little bit if we grew them as biofilms, but they were still quite efficacious. We made 3 cocktails like this, all using the same formula of essentially looking at cross-resistance and then pairing phages that come from different complementarity groups. And we find that this is a very robust way and a straightforward way of making efficacious cocktails. We then put this into a mouse model. So we developed a delayed inoculation wound model where we wound the mice. We give the animals, these are not immunocompromised or anything like that. We just give them 24 hours to form a provisional wound matrix before we inoculate that with bacteria. And then we come in a day later with phage therapy and look after a week to see which mice are still infected. And so when we look at the colony-forming units of bacteria per gram of wound, all the control mice were reliably infected with lots of bacteria. If we gave animals a mocktail of phages from the same complementarity group, some of the animals cleared, but most did not. And then if we gave a cocktail, the KimC1 cocktail or kimchi, most of the animals ended up clearing. So we thought this was quite exciting. This approach works for other bugs. I won't walk through this in the interest of time, but you can essentially do the same thing with Staphylococcus. And here we find instead of 4 complementarity groups, all of the phages we looked at, which admittedly was a bit less, I think it ended up being about 12 phages, fit into 2 complementarity groups. But here again, we find that the cocktails are generally broadly suppressive. I don't have the data here, but it's in the preprint. We looked at complementarity with antibiotics and found that if we combine these with vancomycin and a larger set of Staphylococcus, we get just about 100% suppression with this cocktail. So we were excited about that. Finally, you can actually take the anti-Pseudomonas and the anti-Staph cocktail, and you can bring these together to treat mixed cultures of Staphylococcus and Pseudomonas. And so here again, only the cocktails and not the mocktails worked for suppressing polyclonal cultures. So pulling all this together, I would propose that the palette of tools and workflow that's here, and again, this builds off of many other things in the literature that individually touch on different parts of this workflow, together provide a blueprint or roadmap for making phage therapy cocktails that can be broadly efficacious. I'm sure there's more work to be done with this, but we're excited about the possibility that you could take cocktails like this and combine them in the context of personalized phage therapy or more to the point, a hybrid model where you could develop these as solutions for off-the-shelf phage therapy. And so to pull all this together, I would offer 3 conclusions. The first is that phages that target complementary receptors can produce effective cocktails, that antibiotic classes have characteristic interactions with phage receptor groups, and finally, that the development of off-the-shelf phage therapy cocktails and hybrid models where you're treating patients individually, just like you do in antiretroviral therapy for HIV, but using a suite of off-the-shelf phage therapy cocktails could be helpful for advancing the field. And so with that, I'd like to thank our funders and in particular acknowledge Kevin over here, who did the bulk of this work. He really knocked all of these studies out in about a year, which is pretty phenomenal. And with that, I'll take any questions, and I thank you for your attention.